to this particular endocrine system structure is we're going to start about today is the thyroid gland, which is rather easy to comprehend, rather easy to identify whenever you are identifying this organ or gland in microscopes, under the microscopes. It has got two glands, lobes, right lobe, left lobe, joined together by a tissue, we call them as isthmus. This particular gland has a capsule surrounding to it, and as this capsule surrounding through it, it penetrates deep into it, splits the gland into small lobes and lobules subsequently. So whenever you are going to see this particular piece of the gland, you are going to see some kind of septa which is penetrating deep, just likewise any parenchyma tissue are penetrated by the tissues or of these connective tissues, which you call them as lobules. Inside the lobules, the tissue, we call them as parenchyma. This particular parenchyma is occupied by some kind of tissues or some kind of element. We call them as nodules or colloids or colloidal cells. So this particular gland having a capsule which splits into this particular tissues itself, this particular thing we call them as septus, and this septus goes to split this particular gland into small lobes and lobules subsequently. Inside this lobules, the parenchyma, we find circular round bodies, we call them as follicles. They are kind of circular oval shaped organs, looks like a well. Now this particular follicles is lined by a cells which is known as the follicular cells, and inside this particular follicle it holds some kind of fluid inside, we call them as colloids. So this particular simple architecture exists into this particular gland. You can see this particular schematic presentation how these things looks like. So this is a thyroid gland, and this capsule should be somewhere here. It is projected in the middle of this particular tissue, that's why you are not seeing the septus. Now what we are seeing here, this particular septus which is penetrating deep into it and splitting this particular gland into small lobules. This is a small district which you are seeing here, we call them as nodules. Now within this particular nodules, we find various stage of this various shapes of this particular collections of these parenchymal tissues. We call them as colloids or colloidal body or follicles. So this particular thing we call them as follicles, which is looking like circular element. So follicles may be ovoid, may be circular, which is present in it. It have two components inside. It has a cavity. The central part which is occupied by it, we call them as cavity, we call them as follicular cavity. Surrounding here follicular cavity, we have a lining cells over there which is made by this cuboidal cells to columnar cells, which might have micro border or micro villi into it. So these lining cells we call them as follicular cells. So these particular cells which are cube shaped cells predominantly, sometimes we see that depending on the stage of these tissues may have these particular cells, it will be little bit longitudinal. So that appears as a cuboidal to columnar kind of cells. So this particular element we call them as this colloid which is surrounding by this follicular cells as known as follicular element. Now this particular follicles which is containing the fluid which you are seeing here, we call them as colloidal fluid, which takes the kind of like a pink stain like this, being this acidic nature. Now this particular colloid, depending on how this particular stage or how this particular follicular stage in phase, they have this particular colloidal existence. If the cells are very highly active, that means if the cells are producing too much of like a substance which is coming from it, this colloid should be empty. The emptiness of the colloid, that means the cells or these particular follicles or follicular elements are highly active. If it is filled up with fluid, that means it is sedentary, that means it is not working, it is resting in phase. If it is half filled, half not filled, that means it is partially working, partially not working. So this particular element or thyroid gland produces the hormone, we call them as thyroidal hormones. It has got three such elements which is coming out here, we call them as T3, T4 substance, and in between this particular folloid we have another substance coming in here, we call them as parafollicular cells, which produces a hormone which is known as calcitonin. So this particular element which is present in between there, we find some kind of cells, we call them as parafollicular cells or parafollicular C cells. Now we cannot see these parafollicular cells, C cells, because they are very microscopic, very tiny. We have to go for a super zoom or near to like electron microscopic enhancement to see these cells. they should be present like in between the cells, maybe embedded within this particular follicular element like this, or maybe just like lying in between these follicles like here. So this particular parafollicular cells which should be placed in here is not visible in our magnifications what we are doing here. So these three elements are producing the hormones which is present in here, T3, T4, which is coming from these follicular cells, taking up this particular colloid substance which is predominantly filled up with iodine inside to it, and then this particular element between this particular between the follicles we have a space, parafollicular space, which may have an occupancy of a third kind of cells, we call them as parafollicular C cells, that is also hormone producing cells. So these particular parafollicular cells we cannot see that. Other than this, between these particular capsules, as these capsules are penetrating to these glands, through these capsules we can see the blood vessels running in here. So these blood vessels runs through the septas which is penetrating through these areas and splitting this region. So this region which is separating this particular gland into this particular small lobules, these capsules can be invading in here and making the septas. Within the septas we might find the blood vessels which are present in it. To summarize the thing, it is rather easy architecture that we see. We have a capsule, we have this capsule penetrating deep, and this capsule becomes septa or partitions and split this particular thyroid gland into small areas. We call them as thyroid lobules. Each of the lobules is occupied by the circular bodies, we call them as follicles. Follicles are of two component side. One is the colloid which is holding inside. Now if this colloid is fully present, that cells or follicles are in active stage. If this particular colloid or colloidal space is empty, that means it's highly functioning. Now this particular follicles is lined by the cells which is usually simple cuboidal, cube shaped to low columnar depending on the stage where these particular cells are there. We call them as follicular cells. And apart from that, outside the follicle we might find some kind of cells which is present in interfollicular space. We call them as parafollicular C-shaped cells which you do not see in microscope. That just like leaves us from burden of identifying it. So whenever you are seeing this particular element, this is a schematic picture and this is the zoomed or super zoomed kind of picture. This area where I am rolling the cursor is the colloid which is filled up with. That means this is filled up fully. That means this particular colloid or follicle is in inactive phase. Now if this particular colloid is empty, that means it is highly active. That means it is producing a lot of hormones like T3, T4 which is secreted already. Now other than this half filled, that means partially working as I mentioned. Now this is how it should be looking like. The septas are present on these outer margins which is present in here. As the septas or capsules which is present penetrates deep, we see these small distics are coming, and these are follicles are of different shape or different stages are there depending on the maturities. Now if you zoom this, each of this follicle, it should be looking like that. There will be lining cells which is present in here, cuboidal cells which is lining in here, we call them as colloidal cells, follicular cells, and inside here we have the colloids which is present in it, and outside to it between this particular colloids parafollicular space holds some kind of cells, we call them as parafollicular cells. We are not going to see that, we know that theoretically these particular elements are there. So lobules, follicles, these are the hallmark to identify this particular cells, and this is what we are saying here. If you seeing this colloids looking like this, that means it's in inactive phase. If you see this particular colloid empty, that means it is super active, rather very active. That means it is producing the hormones. It is partially filled, that means it is half working, half not working. So we can have we can assume that the status of that particular follicles, whether it is active or taking rest. In pubertal time, this particular thing is usually empty because that time it is producing a lot of hormones like that. But before puberty, pre-pubertal time, if it is taken up, this particular colloid usually filled up with colloid material. This is high content of iodine which is present in as a colloid substance, and through the colloids this particular follicle cells synthesize the T3, T4 which is liberated into this blood system through these particular blood vessels which is present within the septas, and endocrine functions which is liberated, which is been demonstrated in rest part of this particular body depending on the functions which is going on. So what this particular parafollicular cells are producing, what the follicular cells are producing, that's the exam question. Many times we see parafollicular cells which produces a hormone called calcitonin, The main follicular cells produces T3, T4, and this particular T3, T4 and these follicular cells and all these functional stimuli of this particular thyroid gland is coming from the pituitary gland hormone. We call them as thyroid stimulating hormone. In short, we call them as TSH. So, TSH stimulates this particular status of this particular gland, whether to be active or be inactive, and depending on the stimulation coming from the TSH, this particular follicles are acting appropriately, and so the parafollicular cells. So, whenever you are going to see this particular microscope, 4 of them, they are split amongst groups, see them, you will be able to see this particular easy slide. So, see these particular follicles, follicular cells and these colloids which is present in it. So, some of them may have the septals visible, some of them are projected in the middle part. So, if it is projected in the middle part, you may not see the capsules, but you see the septations which is making a white lines like this and making this particular small nodules. But altogether it is a very easy gland to identify. So, go and check by yourself, take a picture, identify, draw a picture and face off.